Okay, so there I was, wondering what to do for my biology project. After a while, I decided on making a video about how antibodies work. So, here it goes. This is what the average antibody looks like in a diagram. It is essentially a Y-shaped protein, also known as immunoglobulins, which are created by plasma cells, split from specific B cells during an immune response. They are one of the only things capable of neutralizing diseases within the body, aside from macrophages and neutrophils. They are also an integral part of the immune system's defense mechanisms, as it is the reason why we are allowed or we are able to create immunities against these diseases. It consists of 4 polypeptides, making it a quaternary structure protein. The 4 polypeptides can be seen by the red circles shown in the diagram. These are also bonded together by disulfide bonds, as shown here, which all form a single antibody capable of binding to antigens. The circle represents the antigen binding fragment of the antibody. What it does is it binds to the antigen found on the surface of diseases. It works similarly to enzymes which have active sites, or like a lock and a key. Just like how each lock has its own specific key that unlocks it, only specific antibodies have specific antigen that they can bind with in order to carry out functions that help neutralize these diseases. This makes them specific and means that there are a lot of different kinds of antibodies to combat every type of disease. Now, this raises an important question. How can the body respond to all types of diseases with the correct antibodies? Well, the answer isn't so simple. The graph shown here represents the average immune response from the immune system of a healthy person towards a new disease. It is mainly divided into 3 parts: the initial exposure, the primary immune response, and the secondary exposure along with the secondary immune response. After the initial exposure, it takes some time for antibodies to be produced. This is because it takes some time for experimentation for finding the correct lymphocyte that can produce antibodies that fit perfectly with the antigen of the disease. Once these specific lymphocytes are found, they start to replicate under mitosis into memory cells and plasma cells. These plasma cells are the ones that mass produce the antibodies found in the primary immune response. However, the primary immune response only has a low amount of antibodies. This is because, due to the small number of specific lymphocytes, the plasma cells are also lacking in number, and therefore, only a small amount of antibodies can be produced in the initial exposure. However, the memory cells produced during the primary immune response are carried over towards the secondary exposure. The moment they come in contact with the presence of the same disease a second time, these memory cells start replicating rapidly and produces the same plasma cells that are used to mass produce the same antibodies. This is why a lot of plasma cells can be produced at once, and a high number of antibodies can be produced to eliminate the disease at the fastest time possible. In layman terms, we can think of antibodies as ammunition for a weapon, such as the immune system. Ammunition comes in all sizes and forms, just like 9mm rounds, 5.56, or even 45 ACP bullets. But it doesn't have to be restricted to just real-life guns. It can also be, for example, ninja stars, airsoft BBs, or even the Nerf darts that you bought for your nephew last year. Each and every antibody has their own way of fighting off diseases, and here are some important examples. The first type of antibodies we're going to cover are the ones that combine with viruses and bacterial toxins, as shown in the diagram below. Now, to put this into a simpler perspective, think back to the 2015 Tokyo drone wars, where local gangs in Japan transported drugs across the city using flying drones. Think of the drugs as, for example, viruses and bacterial toxins, and the city being our body cells, while the police drones as antibodies. Essentially, what's happening here is the antibodies capture these viruses and bacterial toxins and prevents them from going into the city or our body cells, protecting our cells. The second type of antibodies that we're going to cover are the ones that attach to the flagella of bacteria, as shown in the diagram. However, to find an example for this in real life, you could technically apply the same example of the Tokyo Drone Wars. However, that would be unoriginal, so I thought of a different solution. Instead, let's use the example of tranquilizer darts. The reason why I say tranquilizer darts is because of mainly one thing. These antibodies only work well against bacteria with flagella. Now, if you compare the two diseases from the left and right, you can see that the left one is a bacteria or a living thing that can move with its flagella, while on the right it's a virus that's still and cannot move on its own. Which means that these types of antibodies can only work well against the disease on the left, while when it comes to the disease on the right, it will be ineffective. Now, this is also similar to how a tranquilizer dart can be used to subdue animals or living things that move, while when it comes to inanimate objects, like, let's say, a rock, it won't do much against it because it's already still, and it isn't even living to begin with. The third type of antibodies are the ones that clump bacteria together in a way of agglutination, as shown in the diagram below. Essentially, what they do is they clump bacteria together to immobilize them all and make it easier for the phagocytes or the macrophages to devour them whole. If we think of a real-life example, it would be similar to a net, where one would be immobilized and be left there standing for an arrest or something like that. However, we're talking about multiple targets at once, or multiple bacteria at once, not just a single bacterium. So using this net wouldn't be suitable. Think about the ones in the Pokemon anime where Team Rocket captures a bunch of Pokemon at once. The fourth kind of antibodies is probably the most straightforward kind, that is to punch holes into the cell walls of bacteria. Essentially, how it works is with the help of outside molecules, the antibody is able to create a small hole in the cell wall of the bacteria, causing it to burst due to the outward flow of osmosis due to the difference in water potential. Now, as this is essentially punching a hole into the cell wall, I don't really need to explain how this is similar to real life. It's essentially the same thing as an actual bullet and how bullets work, making holes into places where there shouldn't be holes. Now, the last kind of antibodies are the ones that coat bacteria for easier digestion. How it works is, first, the antibody attaches to the antigens on the surface of the bacteria. Next, it gives out a marking or signal for phagocytes to come closer. Third, the ends of the antibodies are attached to antibody receptors found on the phagocyte membranes. This allows the phagocyte to instantly engulf the bacteria along with the attached antibodies, which makes it easier to consume. Think of this antibody as a flare gun. A flare gun can be used to call a signal for others to for help, just like how in real life you can use a flare to call on an airstrike onto enemies. Now, instead of having missiles raining down on bacteria, it would be just a huge blob that would come towards them and devour them whole, while them being immobilized and not being able to run away. It's similar in a way, but the way of killing or neutralizing the bacteria are completely different. And that is all I have for today. In conclusion, antibodies are an essential part of the immune system's ability to develop immunities towards disease. And it is because of antibodies that we are able to survive as a species up until now. Thank you for watching, and as always, antibodies work.